Am I audible last? Respected examiner sir, all senior professor, Ph.D scholar, master student and other audience. Good morning all of you. I am here to present my Ph.D work entitled In Vitro Studies on Effect of Growth Factor and Oxidative Stress on Human Retinal Pigment Epithelial Cells. The title itself suggests that this is in vitro study on human retinal pigment epithelial cells. When we talk about the structure of human eye, briefly it is composed of cornea, lens and retina. The retina is a very complex structure. For this particular study, we have focused on retinal pigment epithelial cells, which is located between photoreceptors, rods and cone and bruch or bruch membrane. When we talk about the individual retinal pigment epithelial cells in nature, it means that it has an apical side and basal side. Due to polarity in retinal pigment epithelial cells, the protein distribution is also polarized. These retinal pigment epithelial cells plays a very important role in a normal functioning of retina or important in a normal vision. These retinal pigment epithelial cells are monolayer structure which, as I mentioned, plays a very important role from blood retinal barrier to transport of nutrients from inner retina to outer retina. If somehow there is a problem occurs in these retinal pigment epithelial cells, then what? That condition results retinopathy. And retinopathy is nothing but any impairment in the retina that leads to vision loss. For this particular study, we have focused on two types of retinopathy, that is diabetic retinopathy and AMD, that is age-related macular degeneration. From this picture, we can see that the comparison of normal eye with retinopathy condition. Eye with retinopathy condition, here we can see that the degenerated protein, blood hemorrhages, abnormal blood vessel formation and many other things which is not found in the normal retina. From this picture also we can say that this is a comparison of the normal retina with a diabetic retina fundus view. Here we can see that blood hemorrhages, degenerated protein, abnormal blood vessel formation and many other things. When we talk about the impact of diabetic retinopathy at worldwide level, then it is estimated that by year 2030 it is about 200 millions. Now, when we talk about the impact of diabetic retinopathy at India level, then it is about 17%. From this picture, we can see that the comparison of normal human retina with the age-related macular degeneration fundus view of retina. Here we can say that degenerated protein, that is exudates, cotton wool spot, abnormal blood vessel formation. When we talk about the impact of age-related macular degeneration at worldwide level, then it is estimated that by year 2040 it is about to 300 million. This picture explains about the comparison of normal human retina with the two type of condition of AMD, that is dry AMD and wet AMD. In wet AMD, abnormal blood vessel formation can be seen. From this picture we can see that there is a condition of the proliferative vitreoretinopathy. Here the abnormal growth of the retinal pigment epithelial cells occurs, and due to that the condition result that is known as a loss of visual acuity. From this picture we can see the what kind of vision with the patient of the diabetic retinopathy and patient of age-related macular degeneration. The topmost picture explains about the very normal condition. The middle picture, that is DR label picture, explains about the what kind of vision of patient of the diabetic retinopathy condition. The AMD label picture explains about the central cotton wool spot where in case of diabetic retinopathy the scattered spots can be seen. Now the another term that come across us, that is TGF beta 2, or that is transforming growth factor beta 2. The normal level of the TGF beta 2 is very much required in normal functioning of retina and many other organs. But if somehow the condition results where the level of the TGF beta 2 is found increase, then that condition leads to abnormal condition, particularly here in retinal pigment epithelial cells. Elongation of the retinal pigment epithelial cells, which lead to retinopathy condition. In retinopathy condition, it is found too much increase in retina and other surrounding parts. This picture explains about the comparison how the TGF beta 2 help in the normal functioning or normal homeostasis of the other organ, and how if somehow its level increase, then that leads to condition of various disease condition. This picture explains about the TGF beta 2 signaling pathway, which is very much involved in the retinal pigment epithelial cells. Here we can see that canonical signaling pathway of TGF beta 2 and non-canonical signaling pathway in TGF beta 2. The another term that come across us is EMT, that is epithelial mesenchymal transition. It means that conversion of these epithelial cells into mesenchymal cells. For conversion of normal retinal pigment epithelial cells into mesenchymal cells, there are various factors are involved for conversion. For example, TGF beta 2, oxidative stress, and many other. For this particular study, we have focused on TGF beta 2 and hydrogen peroxide, which is oxidative stress inducing substance. And due to that, the normal retinal pigment epithelial cells is converted into mesenchymal cells. From this diagram, we can see that there is a imbalance between the reactive oxygen species and antioxidants, that condition known as oxidative stress. For this particular study, we have utilized hydrogen peroxide as a stress-inducing substance. Earlier TGF beta 2 and here the hydrogen peroxide. For this particular study, we have utilized hydrogen peroxide from the Merck and TGF beta 2 from Peprotec. This picture explains about the involvement of the various antioxidants in the mitigation of the reactive oxygen species in the body. These antioxidants, for example, superoxide dismutase, catalase, glutathione peroxidase, glutathione reductase. So, from literature, we come to know that how and what condition leads to EMT of retinal pigment epithelial cells, or only hydrogen peroxide or TGF beta 2 both are involved in the EMT of retinal pigment epithelial cells or not, or what other condition leads to conversion of the normal retinal pigment epithelial cells into abnormal retinal pigment epithelial cells.
Abnormal cells. Why do you think that hydrogen peroxide plays a role in the environment of the retina?
Yes. In normal retina, there is a normal level found of this particular hydrogen peroxide. In pathogenic condition, we can say that or pathological condition, this level is found too much increase, and due to that, there is a problem occurs in retina. That's why we have utilized that H22, and ultimately how oxidative stress is involved in conversion of EMT of retinal pigment epithelial cells, that we have tried to know. So what we hypothesize, what is role of the hydrogen peroxide or what is role of oxidative stress in conversion of the normal retinal pigment epithelial cells into abnormal or mesenchymal retinal pigment epithelial cells. So our aim to understand or to evaluate the role of hydrogen peroxide and TGF beta 2 in conversion of the normal retinal pigment epithelial cells into abnormal retinal pigment epithelial cells. For this study, we have utilized ARP19 cells, that is, we have utilized from ATCC USA, and all these are condition of the cell culture condition. For this particular study, we have utilized various cell culture method for completing or for getting results and for experiments. These are the different methods we have utilized that I'll explain along with the result part. To understand the entire result part, we should know that what are the different groups we have utilized for this particular study. From control, only TGF beta 2, only hydrogen peroxide, and combination of the TGF beta 2 and hydrogen peroxide. In this picture we can see that this is explained about the cell survival condition. From graph we can see that as the concentration of the hydrogen peroxide increases, the cell survival decreases because it is a stress inducing substance. From this picture we can see that this is a result of effect of TGF beta 2 on cell survival of the retinal pigment epithelial cell line, that is ARP19, and this is growth factor, so it does not show any cytotoxic effect. From this picture we can see that the treatment of the combination of the TGF beta 2 and hydrogen peroxide on a ARP19 cell line. From here we can see that at the low level of the H2O2 is given along with the TGF beta 2, it does not show that much cytotoxic effect, but it is given along with the TGF beta 2, it shows cytotoxic effect. From this picture we can see that this is very normal cell morphology of the ARP19 cell. When it is, from this picture we can see that there is, when it is given along with the TGF beta 2 and hydrogen peroxide, it shows very most elongated cells. It means that retinal pigment epithelial cell morphology changes, it becomes more elongated. It means that EMT. This picture explains about the cell migration assay representative images. From these cell migration pictures we have prepared one graph, and that graph explains that when the combination of the TGF beta 2 and hydrogen peroxide given, results are suggested that when it is given in combination, cell migration of the ARP19 cells increases. It means that EMT of retinal pigment epithelial cell line ARP19 cells occurs, and that all occurs due to oxidative stress condition. This picture explains about the reactive oxygen species measurement. As we are talking about this oxidative stress condition result, so for that we have utilized DHR123 that measures the total reactive oxygen species in the particular cell. So we are talking about the oxidative stress condition when it is given the treatment of the TGF beta 2 and hydrogen peroxide. Researchers use different markers, epithelial markers, mesenchymal markers or EMT markers to determine the alteration in a change in a morphology of the retinal pigment epithelial cells. This picture explains about the disintegration of the tight junction protein ZO1. So from this lowermost picture, when we compare with the normal ARP19 cells, then we can see that delocalization of the ZO1 protein. It means that from the outer part or from cell junctions, that ZO1 disintegrate and transfer into the cytoplasma. So disintegration of the junction protein results, and that is due to oxidative stress. In another way also we can say that the fibronectin protein, which is marker of the mesenchymal cells, or it is EMT markers. So increased fibronectin, or here these particular pictures, increased fluorescence is directly proportional to the amount of protein. So the fibronectin protein is found increased. It means that the cells are converted into mesenchymal cells. These diagrams and pictures and results explain about the protein level markers. We have, I mentioned that ZO1 and fibronectin were utilized as a marker of EMT markers. So at protein levels, the same results we have confirmed. The increase fibronectin and decrease the level of ZO1, that is a junction protein. These markers are also checked at mRNA level, the epithelial marker, that is ZO1, and mesenchymal markers and along with the other marker also, that is a fibronectin and many other, where we found that the EMT occurs in the retinal pigment epithelial cells. This graph and picture explain about the level of glutathione or total glutathione content in the treatment of the TGF-beta 2 and hydrogen peroxide, where we found that at higher level of the hydrogen peroxide there is a higher oxidative stress can be seen. This picture explains about the antioxidant enzymes activity measurement, where we found the oxidative stress condition increase.
Have you checked the level of glucose DSSP in oxidized glucose?
No, sir. We have not.
That's very reduced level of glucose.
So we
You
have
have
not.
not checked the oxidized glucose.
No, sir.
Because when you check,
Okay.
that will show what exactly happens. Because that level should be more, make it more oxidized.
Okay. May I continue, sir?
This is the tabular representation of the in silico analysis, so better we see in that particular diagrammatic view. So this diagram explains.
Here in this picture we can see the different.
What kind of insulin?
Okay. Okay. So we have utilized CAG pathway enrichment analysis. So by that actually there are different softwares are available for doing that actually. So based on that we have prepared this table, and here in this. Okay. So based on how that oxidative stress is related with the diabetic retinopathy, how oxidative stress is related with the PVR, that is proliferative retinopathy, and how oxidative stress is related to glutathione. So what are the different proteins are involved in this particular disease condition? Based on that we have, these are the list actually, different proteins which are involved in this particular disease. So this picture explains about the oxidative stress and diabetic. So that much proteins are involved in something, there is a problem occurring in retinal epithelial cells. Out of which the highlighted proteins we have confirmed at our level only in this particular study. This picture explains about the B and C, that is relation of the oxidative stress with PVR and relation of the oxidative stress with glutathione. These are the highlighted proteins we have studied at our level.
Why did you list that hypoxia insulin factor? That is more important in both AHMEDABAD and retinopathy. Because hypoxia is one of the factors causing proliferative retinopathy. So why that factor did not be have not been selected? But it is not selected. All the proteins.
So based on this in silico analysis, we just come to know that based on in silico analysis, we just come to know that these are the different proteins are there. Based on that, we also prepare this diagram, and these are the different signaling pathway are involved in this particular conversion of the retinal pigment epithelial cells into mesenchymal cells. So at signaling level, we haven't confirmed. On the in silico study, we have only utilized to just understand that how and what different signaling pathways are involved. That much we have. That's why we have only utilized in silico analysis. These different signaling level we haven't worked in this particular study. So here I would.
If you had to do this, understand what you're talking about. If you had to check the signaling level,
Yes, sir.
what can you do?
We need to focus on different particular proteins.
What do you do? If you want to know what is the signaling pathway,
We we have to check the protein levels.
Which marker? Which pathway?
Okay. We have to focus because currently we are talking about the TGA beta, most important which is involved. So we have earlier picture I have explained there is a two signaling pathway, particularly canonical pathway and non-canonical pathway are involved. So different proteins are there, different complex cascade is there. But we have to select the proteins and we have to check the protein level and particular protein we have to. Okay, definitely, sir. Yes, sir. Okay, sir. Definitely, sir. Thank you.
Thus, we may conclude that when treatment of the H2O2 and TGF-beta given, there is a oxidative stress condition results, and here the reduced glutathione can be seen. It means that due to oxidative stress, the normal retinal pigment epithelial cells is converted into mesenchymal cells due to EMT, and as we have discussed, there various factors are involved in the EMT of retinal pigment epithelial cells. From in silico analysis, we have just come to know that various complex proteins are involved or different signaling cascades are involved in the conversion of epithelial cells into mesenchymal cells, and we have utilized in vitro analysis to understand how and different markers are involved in the conversion of retinal pigment epithelial cells into mesenchymal cells.
Your question is from here, so actually we haven't utilized actual signaling pathway. So definitely if we
The question was if you had to do it,
okay,
what kind of
definitely if we get chance then we'll
proteins
okay, and that the answer is phosphorylated protein we have to talk about. Check. Thank you, sir.
So when we're talking about the future prospect of this particular study, then yes, based on this study, we just come to know, or we have just utilized in vitro analysis for understanding the results. But in future, we will incorporate in vivo analysis where we will include human retinal pigment, primary human retinal pigment epithelial cells. Further, we will try to work on a mice level and other higher in vivo analysis. Some of the markers in this particular study we have skipped, so we'll try to incorporate that marker also. Particularly your suggestion, that is a phosphorylation of this particular different TGF-beta signaling pathway or marker protein, that also we will incorporate. Further, some of the anti-EMT agents, it can be phytochemicals or it may be other agents, can be utilized. So these are some of the references that I have utilized for particular this presentation, and these are some of the from this particular study. Very first publication from my side, but that is as a co-author. In this article, we have discussed how the involvement of the EMT of retinal pigment epithelial cells. In this particular article, we have discussed about the role of antioxidant status in the retinal pigment epithelial cells. In this particular article, we have discussed about the role of the lipid peroxidation in the EMT of retinal pigment epithelial cells. In this particular article, we have discussed about the different signaling cascades which are involved in the EMT of retinal pigment epithelial cell. In this article, we have discussed about the role of combination of the hydrogen peroxide and TGF-beta. In this particular article, we have discussed about the role of AMD in a retinopathy and its treatment strategy. These are some of the other articles as a co-author. All other are the abstract that I have published in a different conferences. Here in this particular step, I would like to mention that I have given one of the international talk in the international conference. These all are different publication. Here I would also like to mention that during my PhD tenure I got second prize in a PhD category in one of the national conference. This is the list of the different conference. Here I would like to thank my PhD guide Dr. Kite Johar for his contribution. Here I would like to thank examiner sir. Further, I would like to thank all the senior professors and Ph.D. scholars from Department of Zoology, Gujarat University, Ahmedabad for their support. Further, I would like to thank all the non-teaching staff from the Department of Zoology, Gujarat University, Ahmedabad for their support. Further, I would like to thank Gujarat University for considering me as or giving a platform as a PhD scholar in Gujarat University. Now I think this session is open for question and answer.
So the presentation is open for questions. You can ask questions. Please. So from students, let's see.
And finish one question.
Okay. So where is the source of hydrogen peroxide? So where is the source of hydrogen peroxide?
Question is very specific, sir, but there are various metabolic activities are going on. So to regulate the reactive oxygen species, there are various antioxidants are there. So, okay.
So your problem is the hydrogen peroxide is your problem.
Okay.
I am asking where from the hydrogen peroxide generates. What is the source of hydrogen peroxide?
There are various metabolic activity, but very specifically, currently I am not able to answer.
So any idea about mitochondria?
Definitely, sir, it is like that.
What is the mechanism in mitochondria?
Yes.
What happens?
Yes, sir. Exactly.
There is a gap of 2 years after submitting the thesis. Any other question from audience? Chalo, please. कोई question नहीं? बस समझ में ही नहीं है। या तो सब कुछ समझा या तो सब नहीं। So now I invite Professor Vivek Tanawde to hear his question, and the other evaluator of the Vijay's thesis was Dr. Bhanu Prakash Reddy from National Institute of Nutrition, NIN, Hyderabad. And I have made sure that he incorporates all of these into his thesis. He has not written any question to be asked, but that, sir, that you can decide and you can ask the question.
Direction. You said that your last objective was that if you have some agents that can inhibit oxidation,
Yes.
and you want to see their effect on EM, but for your hypothesis to be proven, you would also need to measure their effect on TGF beta itself.
Definitely.
Because what you are saying is H2O2 is contributing to EMP through TGF beta signal.
Okay, definitely.
And so when you or you or anybody else designs the experiment where you have antioxidants,
Yes.
and you want to look at the effect of this on EMP, you also have to study what's happening with TGF.
Definitely. Yes.
We recommend that you be awarded the PhD degree. So So that's congratulations, Dr.